I'll be delivering this session on exploring genomics industry opportunities and innovations. So this is mainly focusing on our this young minds who are pursuing post graduation studies in different disciplines. So the flow of the session would be like this. So we can have an introduction session on genomics, then that is followed by overview of the genomics industry. So during that, initial speakers also were mentioning about the this industry opportunities and lack of the knowledge on this industry. So that also we can discuss what are the industrial opportunities available in this genomics and how can the students be ready for that industry. Then we can discuss on the scope of genomic industry. So that is whenever we look for an industrial job, we check whether there is a scope for this further growth or this further career opportunities. So that also we can discuss in this session. Then we can have this briefing on clinical and consumer genomics. Then genomics testing technology, what are the technologies we use at our company to develop our products, and finally what are the genomics opportunities at OhMyGene. So that means actually how the students of University of Kerala approach us for an industry academia collaboration. The briefing of that I'll be giving, and the more detailing of that collaborative activity Dr. Gauri will be speaking. Then let us go to the first part, the genomics, an introduction. So as we know, this genomics, when we talk about genomics, genomics is the study of an organism's entire genome. So we all know what is genetics. When we talk about genetics, we speak about one gene or the abnormalities of a single gene, whether that is causing some clinical outcome or clinical significance is associated with that one. So earlier days, we used to test one gene or two genes to check whether there is a clinical implication of that one. But nowadays, due to the advancement of technologies, especially the next generation sequencing technologies, we can sequence the entire human genome, and we can see whether there are any defect present at any of the genes or the chromosomal regions. So looking at that whole perspective, that is the genomics, that is looking at the entire genome of an organism. So there are a lot of applications also for this one, and so when we study or when we use this genomics, we can utilize this technology for various aspects. All those aspects we will be discussing in the coming slides. And in between, so if you have any questions in between, we can discuss that. You can interrupt me in between. Otherwise, you can reserve the questions at the end of the session. So either of that would be fine with me. Okay, so as I mentioned, the technological advancement, that was the one of the keys for the flourishing of this genomics industry. So when we talk about the technological advancement, so we know the PCR polymerase chain reaction, and also starting from that, then the Sanger sequencing came this into place, and through the Sanger sequencing, we could sequence a part of the gene or there's a particular length of a gene. Then from there onwards, that is during the 2002, that is the beginning of the 2000s. So the sequencing technology was there. Then later, lot of advancements happened in the sequencing technology. And now when we are here in 2024, so we have the technologies, very fast technologies. Utilizing that, we can sequence the entire genome. So there are a lot of key players also in developing this technology, starting from Thermo Fisher Scientific to Illumina to MGI and so on. And there are different technologies also these people are adopting. So mainly this sequencing, we can classifying into two main types: sequencing by synthesis, and also this companies like Illumina, MGI, they are focusing on the sequencing by synthesis method. And there is another technology that is developed by Ion Torrent. So they measure the H+ ions released when the one base is attaching to the strand. So then they measure the H plus ions, and based on that they make the this determination what is that base and all. So that is the ion torrent technology. So likewise there are n number of technologies and technological advancements happened in this field of genomics. So because of that contributed a lot to this genomics industry. And not only the technological advancements, initially the cost was an issue. The cost means actually when they started, the cost of sequencing was this $100 million. So we know now that, so if we want to go to space, we need to spend $100 million right now. So because of that, that is not a dream for this everyone. Only a few people can this afford that. When this genomics technology started, that is in 2000s, so the cost of this entire human genome sequencing was $100 million. So only a very limited this folks could afford that, and that was kind of a this very dream for others. But due to the advancement of technology, so this technological advancements also follow Moore's law. Moore's law means, so in every 2 years, there is a doubling of the computational speed, because all these are this big data based analysis. The computational technology or computational advancements are required to analyze the data. So when this technology is advancing or the computational power is doubling, so there is a reduction of the cost. So likewise, it started with 100 million US dollars in 2000. Now, so for a few $1,000, or we can sequence the entire human genome. So that is where we are now. So this is the advancement happened in the last 20 years. So these two are the contributing factors of this affordability of this genomics. And also then researchers, they started exploring this genomics to make it beneficial in the this clinical genomics, or in the agricultural sector, so or in the this other drug discovery sector, for pharmaceutical sector and all. So yeah, so with this, actually, I think I set up the introduction of this genomics, and now we can go to the overview of the genomics industry. So how this genomics industry is flourishing now, and who are the key players of this genomics industry. So when we look at the global genomics market, so now this is the data from 2022. So the global genomics market was 44.6 US dollars in billions. So there is a compound annual growth rate of 12.4%. So that means, so based on this compound annual growth rate, there is a prediction that by 2028, the global genomics market will attain 83.1 billion US dollars. So if by seeing this representation, you can imagine what is the scope of genomics market. This is a global market, and this red one, that is showing the market from North America, and this blue one, that is the European market, and this orange one, this is the Asia Pacific market, that is where India comes, and then Latin America and the Middle East. Even in the Middle East also, so they are this expanding in this genomics. They are having a lot of collaborative activities with the companies like NGI and all, and they are expanding. So this why I included this picture is that, so this global genomics market is giving will be giving you that notion that there is a scope for this genomics worldwide. So so we know that there are only limited options in Kerala, but there are a lot of huge opportunities huge opportunities for genomics worldwide. And so if you can also dream, every one of you, so that all these postgraduate students, you can also dream big and achieve big goals. So if you pursue genomics. So Let me also discuss the ecosystem analysis. So what are the different domains coming under this genomics, and so what are the opportunities for people, especially this here also you are from various disciplines like the zoology, biotechnology, bioinformatics, botany and all. So for each one of you, so you is what based on your skill set, you can also decide which area to be focused. So genomics is a very broad term we use for all these ecosystems. So depending on the skill set, you can also choose the area of interest, and you can also build your skill set to be industry ready. So coming to this product and services based this ecosystem analysis. So now people are just developing automated genomic systems. But for us, actually, this will be very this will be wondered by hearing this automated genomic systems. Am I audible?
Yes,
Yes,
ma'am.
madam.
Yes, yes, yes. Yes, ma'am, audible.
Oh, okay, okay. I think I got a message that somebody muted my presentation. Okay, so let me continue. Okay, so this now in developed countries, people are working on this automation on this genomic systems. Automation in the sense actually for so in genomics there are wet lab part and the dry lab part. Wet lab part means actually that's very laborious. This experiments are required for this genomics, for especially for this next generation sequencing. So this whenever this people are doing or researchers are doing, there is a chance of error. So to avoid all those chances, error chances, actually people are this automating all these systems. So that much advancement has happened or happening in this field. Then this test kit development and reagents, as I mentioned, so there are a lot of this laborious this experiments required for this one, especially for library preparation and all. There are requirement for reagents and test kits. So there are these companies working on developing these test kits and reagents, and also this huge computational this power and analysis is required for this next generation sequencing or genomics applications. So if a person, if you are skilled in bioinformatics or this software part, then you can also this opt genomics as a career by opting this specific area. Then there are genomic service providers. So that means actually there are specialized skill set required for analyzing the output coming from the sequencing machine. So for that, so there are these companies, they provide only the genomic services. That means they won't be having a huge laboratory setup, but they will be having tools and this softwares for analyzing their data and giving the interpretation and the reports. Then actually these are the this basic things that is you might be knowing about this polymerase chain reaction, Sanger sequencing and all. So still actually whenever we do this next generation or whole genome sequencing, so there may be some confirmation required for to test the presence of some variants or mutation in one or a set of genes. So in that case, this real-time PCR based assays or Sanger sequencing based test will be this followed to provide the confirmatory results. And there is another this service called microarray systems. Microarray systems means actually this is the just before the this emergence of next generation sequencing, this microarray technology was there. Microarray technology means actually the difference between these two is that in microarray we will be able to identify those genes, those which are known to us, because that is based on the probes. So based on the knowledge of the existing genes, probes will be developed, and those probes will be utilized to check whether that associated genes are present in the sample. So that is the microarray technology. So these are the various others products and services present in this genomics industry. And coming to the application side, so all these genomics services or products which we discussed have applications in diagnostic field, precision medicine, drug discovery, agri-genomics, animal research, and so on. So this major applications we will be discussing in the coming slides. And the technology, we already discussed this technologies here itself. And the end users of these technologies or this ecosystem will be this clinics, even this researchers, then pharma companies, or others in marine research, animal research, or plant research. They will be the end users for this genomics technologies. Okay, now, as I mentioned you earlier, so we can look at the scope of the genomic industry. So even though we talk a lot about genomic industry, so we need to check whether there is a this growth scope, and if we opt the career in genomics industry, what are the scope? So for that, so this is a table showing some metrics. So this metrics, based on this metrics, it is very clear that so there is a good scope for this genomic industry. So let us see this metrics. That is, I have already shown you a graph that was showing the market revenue of genomics industry in 2023. So that was $46.2 billion. Then the projected revenue based on a compound annual growth rate of 12.4%, that is in 2028, that would be $83.1 billion. And the major market driver for this industry is the increased government funding to support genomics project. So increased government funding, you might be wondering. So here we know that in India, we are getting very less funding for projects, but in countries like US and UK, so they get this huge amount of funding, and there are a lot of this specific funding program specifically for this genomics development. So that is the major this driver for this genomics industry. Not only that, this pharma companies also, the private companies also this do their own this research and product development, that also contributes to this genomics industry. And main market opportunity for this genomic industry is in the use of genomic specialized personalized medicine. So personalized medicine, that was a very unfamiliar term for us before, but nowadays this term is becoming familiar to us as well, because when we go to hospital, sometimes we also get this doctor's reference for this test. So most of these doctors, especially from super specialty and multi-specialty hospitals, so they suggest to undergo a genomics test so they can also guide the patient in this personalized way. That is, the personalized treatment or drugs can be selected to have a better treatment outcome. So if we categorize the genomics market, we can classify this in various ways. So I have shown here the 4 different types of classification. That is, by technology, this genomics market can be classified into sequencing-based genomics market, microarray-based, PCR-based, flow cytometry, in situ hybridization, and immunohistochemistry. The last 3 techniques that are this not directly coming into this genomics, but as I mentioned earlier, for some confirmatory test. So still in industry, we are using this techniques like flow cytometry, in situ hybridization, and immunohistochemistry. And by application, we can classify this genomics market into drug discovery and development, diagnostics, agriculture, animal research, marine research, and so on. And by study type also, we can classify this functional genomics. So mainly that is transcriptomics and RNA-seq technologies are coming under this functional genomics, where we correlate the expression or the variation of a gene with the clinical significance. Then biomarker discovery, this is also very much this flourishing area where this is helpful in the diagnosis as well as also helpful in this selection of drugs. And epigenomics, structural genomics, comparative genomics are the other types of other categories coming under this study type categorization. And by end user, so we can classify that this genomics used by hospitals and clinics, genomics used by academics for research purposes, and research institutes, they also develop products based on this genomics. And pharmaceutical industry, they this discover drugs as well as biomarkers, so that is coming under the pharmaceutical category. Then also biotech companies, they use for various applications including health, wellness, and agriculture. Hope you will be able to follow this presentation, and if there are any questions, please do ask. Can I continue?
Yes, ma'am.
Yeah. So we discussed about this categorization of this genomics market, and we focus at our organization, at our industry, that is OhMyGene. We mainly focus on functional genomics. So in functional genomics, under that functional genomics, we use three main technologies like next generation sequencing, microarray, and real-time PCR. Under this next generation sequencing, we use whole exome sequencing or clinical exome sequencing. So we rarely use whole genome sequencing. So that is a laborious activity, and also the cost of that next whole genome sequencing is at the higher end. So mostly we get most of the answers through a whole exome sequencing and clinical exome sequencing. So whole exome sequencing means actually we know that there are this billions of genes present present in our DNA sequence, and out of that, there are 20 to 22,000 coding genes. So whole exome sequencing means we look at only the coding part of the genome, and in that means actually we will be focusing on this 20 to 22,000 genes. And clinical exome means actually that is depending on our this objective. Suppose if we have to identify genes, so which are from a selected number of genes or that is from a particular region of the DNA. So we develop specific panels or select the required genes that is to be tested. So that might be some ranging from 6,000 to 8,000 genes. So that is categorized as clinical exome sequencing. So in our company, we mainly focus on these two, whole exome sequencing and clinical exome sequencing. And we also this depend on microarray, that is mainly for SNB microarray. So in that SNB microarray also, that is based on the requirement. So if we have to check for a specific panel for a particular disease called cancer or some neurodegenerative condition, we know what are the genes acting there. So we make specific panels for that one and design probes to test all those genes. So that is the microarray based SNB test. And this real-time PCR, suppose if we have to test only one or few genes, then we go for the real-time PCR based assays. Then the applications for these technologies at our this company is that mainly we do SNP analysis, single nucleotide polymorphism analysis, and mutation analysis. That is, mutation analysis means if there is a known change that is there in the genome of an individual, and we study the clinical implication of that mutation, and we report that mutation so that the person will get that an indication that this gene is mutated and that he has to take precautionary action or the treatment based on that mutation. Then we also do gene expression analysis. So due to this polymorphism or mutation variation, so there could be a difference in expression of some genes. So we do analyze such type of gene expressions and also correlate clinically, so that medical practitioners or healthcare practitioners will be able to guide that this patient properly or in this personalized way. So we have this products mainly under these two categories, that is clinical genomics category and consumer genomics category. So we'll be discussing what is this clinical genomics and what is this consumer genomics. Yeah. So, the difference between consumer genomics and clinical genomics is that, so we know this, this is similar to over-the-counter medicine and this prescription drugs. That is over-the-counter medicine, we can go directly and this purchase from this drugstore. But for this prescription-based drugs, we need to have this prescription from the doctor. Then only we will get that medicine from the this authorized this drug dealers. So similarly, consumer genomics means actually, so if you want to know, if a person wants to know about his genetic predisposition, if he has some genetic predisposition to some of the indications, or he just as out of curiosity, if he wants to know his ancestry related things, he can opt for this consumer genomics. For this one, there is no need of a prescription or this reference from the doctor. So private companies offer this service. In India, so the consumer genomics market is very less, but in Western countries, there are this huge market for this consumer genomics. And this clinical genomics, as I mentioned, so when a patient is approaching a doctor, then doctor might feel that actually this person should get tested for this particular this genomics assay or some particular panel. Then this doctor suggests this genomics test so that that person can go to a company offering this service, or there will be this clinic industry collaborations, so that the doctor can direct that person to that this clinic genomics laboratory, so that the sample will get tested there, and the doctor will be this receiving the feedback through report, and so that the doctor can decide on his treatment. So that is the clinical genomics part. So at our company, we have both these services, consumer genomics as well as clinical genomics. So in this slide, actually there are two schematic representation. The left-hand side schematic representation, that is the workflow of this clinical genomics activity. So here you can see that actually, as I told you, when a patient is visiting a healthcare practitioner, if that person, doctor is feeling that okay, he has some hereditary associated this genetic disorder, or maybe if he has a cancer, he has a family history of cancer, so then the doctor might suggest genomics testing to check whether he has some variation or polymorphisms in his gene, or some deletions or some kind of a genetic rearrangement happened in his genome. So then that doctor suggests this genomics testing, clinical genomics testing. So then there is he actually, before going to this clinical genomics testing, so there is a person between this doctor and this patient, that is the genetic counselor. So this genetic counselor will be this giving all the information regarding the genetic test, what kind of a test is this one, and that person, genetic counselor, will be asking a lot of questions about his family, about that patient's family, and he will be drawing family history chart like this, and that genetic counselor will be suggesting which clinical test would be suitable for that person. So based on that information, the patient can approach the lab, or this through this genetic counselor, this lab test can be finalized, and the sample can be sent to the lab for sequencing, next generation based or microarray based testing, and the output will be sent to this genome analysis, and the final report, that is report means actually what are the variations present in that person's genome, that report will be generated, and again that report will not be this easily readable to the patient, for this healthcare practitioner. So for that, again, the role of genetic counselor will be there. That genetic counselor will be this telling the details of all this testing and what are the this implications, what are the clinical significance of the variations, if there are any variations identified in that report. Then also that this is called genetic counseling, because actually by seeing all these things, actually the patient may get may go to a panic situation. To avoid such situations, this genetic counselor will be this giving the patient proper advice and what to do and what not to do, everything. And through this one, then actually and with this report, this patient can this again go back to the doctor, and this based on the report, this doctor can decide whether some particular treatment is to be given. Because actually if the problem is there in the genes, all the treatment may not be effective to that person. So then the doctor needs to decide which treatment will be more effective to the patient. So based on that, the doctor will give him this proper advice and also proper treatment. So this is the whole cycle of the clinical genomics. And this also has another section called the pharmacogenomics. So pharmacogenomics means, even though actually we have all these 23 pairs of chromosomes, so we are very much different genetically. So this thing, the one drug which is there for one disease may not be effective for this or everyone in the population. So suppose if there is a particular mutation in a certain subset, then the particular drug will be effective for that person, effective for that subsection of the population. Otherwise, another drug would be effective for the subsection of the population. So to identify that, this genomics testing or this variation testing will be helpful. So and this is called the this personalized treatment also. So the branch dealing with this one, that is called the pharmacogenomics. So this is also, this has lot of this advancements nowadays, and actually there are a lot of side effects can be avoided if we know our genetic nature also. Even not only for allopathic medicine, even for Ayurvedic treatments also, actually this people are adopting, or these practitioners are adopting this pharmacogenomics technique. So that is actually in Ayurveda also, many people, we all heard about this Prakriti based analysis and all. That is Ayurveda is like kind of a personalized medicine. That is what these practitioners are saying. But scientific correlation was difficult earlier days, but with the advent of this genomics and all, now people are aware that, so yes, actually the one medicine is not suitable for all. So based on your genetic nature, you can also opt, or the doctor can prescribe the proper medicine for you. That is personalized approach can be adopted to have a better this treatment outcome. Then here actually I have listed some well-known companies or top companies in clinical genomics market. This is the Indian scenario. So I haven't included this global market here. In globally, there are many players in this clinical genomics market. But if we look at the India, in here also we have many these companies offering this clinical genomics services. The major companies include MedGenome. This is based now we have in India. They have this laboratory in Bangalore. This head office of this company is in California. And then this Newburgh is there, that is based at Gujarat. Then Eurofins Labs, Xcode, Strand Life Science, again this is from Bangalore. Then for Base Care, LifeCell, OhMyGene. So these are these companies offering clinical genomics outside Kerala. So in Kerala, OhMyGene, so that is our company is providing this clinical genomics services. And as I discussed earlier, also there are a lot of applications in clinical genomics. So not only in this healthcare, but also in this other fields also, the applications, genomics application we discussed. Here, this slide is specifically for this clinical genomics application. So for this disease diagnosis, personalized medicine, we have already explained this personalized medicine. And risk assessment. Risk assessment means actually we know this genetic carriers, heterozygous, homozygous conditions and all. So so whether we are carriers, whether we have a risk for developing a disease, so to analyze, to check that in advance, this genomics is very helpful. For example, we know that many people, they do check the BRCA1, BRCA2 mutations for cancer to see whether they are genetically predisposed to that condition. So that not only for this breast cancer, many other cancer, many other this genetic this disorders, we can adopt this genomics testing, and we can also get the information on the genetic predisposition to various indications, so that we can also plan our this lifestyle, or we can manage the lifestyle uh, uh, to have a better quality of life. And also for example, if there is a need for starting the surveillance from early days, that also can be decided based on all this genomics. So that is why so there is an application in risk assessment, and as we discussed, application in pharmacogenomics is there. Then carrier screening, that is as I mentioned. So if suppose if a couple is there, they are planning their family, so they can see whether they are carriers of some indications or genetic disorders. So they can check whether that will be this carried over to the next generation. So there are a lot of these carriers screening for a lot of genetic disorders available nowadays, so that they can also take all the precautionary measures for that. Then again, the prenatal testing. So that is so during first trimester, most of this pregnant ladies, they undergo for this prenatal testing. So that is to check whether there are any genetic associated disabilities in this offspring. And also for rare disease diagnosis. And sometimes actually these rare diseases that manifest after a certain age, like in adolescence or in this after 30 years or so on. So, but you can, through this genomics, you can check whether you have a genetic predisposition to such rare disease also. So that all these will help in your this betterment of life. Then coming to the now actually we discussed a whole lot about this clinical genomics. Now coming to the consumer genomics. So here this is the global picture of this consumer genomics market. So in India there are very few companies offering this consumer genomics. So all these are from this global side: GSK, 23andMe, Gene by Gene, Color, Futura Genetics, AstraZeneca. These are this pharmaceutical companies. They also are into this kind of this genomics industries. And from India, there are this two key players, and that is OhMyGene. This is a Bangalore-based company, and OhMyGene, that is our company. Okay, so coming to the application of consumer genomics. So here, as I mentioned, so this is like a person can decide whether to go for this consumer genomics or not. So as I mentioned, many people, they go out of curiosity to check their ancestry, whether what is their this origin. So in India also there are a lot of studies conducted, like Aryan origin, Dravidian origin, like that. So to know your ancestry, you can go for this consumer genomics testing. Out of other than that, so there are a lot of this very good applications also for this consumer genomics. This mainly in the health and disease risk assessment, as I mentioned earlier. So whether you have a genetic predisposition to certain conditions, genetic conditions, you can undergo. So you can so if you want to know what is your genetic predisposition, and how to plan your this lifestyle, and this how to go ahead with your this future life, you can go for this consumer genomics testing. And again for pharmacogenomics. So but nowadays in India, especially in Kerala, this pharmacogenomics field is not that well flourished. But if you want to check whether many of the people that we are having this lifestyle disorders after 40 years, but we sometimes we also will be confused whether to have all these medicine together, whether this will be cross reacting, whether this will be effective and all. So to have a better idea of that one, to take a decision, so we can also go for this pharmacogenomics, and with that report you can even approach your healthcare this provider and show this result to them, saying that so my genetic condition or I have a genetic predisposition for so and so conditions or so and so drugs. So can you this schedule my this treatment accordingly? So in that way also we can suggest to our healthcare practitioner. Then other application is in nutritional genomics. That is actually we know that some people are allergic or to some food, like there are lactose intolerance, or some people when they eat less fat, they gain weight like that. So that is all those due to your genetics. So if you get an idea about your genetics, you will also get to know what to eat, what not to eat. So that is why mainly people go for this consumer-based genomics testing. Then also this has application in fitness and lifestyle optimization. So how that is possible is that actually there are a lot of traits so related to fitness. So like this endurance, this energy, activity, or this muscle strength, or injury, prone to injury. So like that, lot of traits are there. So through this single nucleotide polymorphism, through consumer genomics, we analyze whether this you have that kind of a genotype, or what type of a genotype you have, whether you are this prone to such illnesses which are associated with this fitness. Suppose if you go for some specific gym activity, whether that is suitable to you or not. So all those information you can have from this genomics testing. And finally, for carrier screening, like as I mentioned earlier, so whether to know whether you have some you are carrying some mutations for some genetic condition, whether that will be transferred to your this next generation. So for that assessment also, one can opt this consumer genomics. And coming to this, our this consumer genomics based test. So we have this if you this consumer genomics based products. One of our this premium product is OMG Genestrology. So this genestrology means actually in the name itself, this is kind of astrology. That means your gene will that is tell you, or the pattern of your genotype will tell you what is your this future, or what is your genetic predisposition. So earlier we had this test based on the saliva sample, but right now this we use a blood sample for testing. So blood sample will be collected from the individual, and that will be sent for this NGS based testing. Right now we do not have an in-house facility for next generation sequencing. We have this partnership with this other companies who have this facility, and we are doing this testing through them. And we have our in-house pipeline to analyze all this data, and through our in-house pipeline we analyze the data and we give the report, and along with that report we provide the genetic counseling services as well. And so if there are some this major problems in that, we also give the genetic counselor also will give a recommendation, so that with this report that person can approach a healthcare practitioner. So our other products are this OMG CAN. So this is basically for this cancer panel, hereditary cancer panel testing. Suppose, like I mentioned, BRCA1, BRCA2, and like that. So there are many genes, many known genes. So that means whether there are some mutations associated with that genes. So that means you have a genetic predisposition to that cancer. So to test that one, suppose if there is someone in your family this who has a cancer diagnosed, and so then you can also see whether you have that chance or genetic predisposition for cancer, so that actually you can also take precautionary measures well in advance. So that is the OMG CAN test. This OMG key means actually this is a subsidiary of genestrology. In genestrology, we test 200 plus conditions plus pharmacogenomics, and OMG key. So we test for this selected number of conditions, and this OMG genestrology, that is an NGS based test. Here actually we provide both NGS as well as microarray based test, as that is a testing a selected number of genes. And this is another product, that is OMG Fit. So as I mentioned earlier in the earlier slide, that is this assess your fitness based traits. That is, what are the polymorphisms associated with your this fitness traits, and what type of an exercise or what type of this physical activity will be suitable for you. So this test is mainly for that. And we do have this clinical genomics based test also. So again, as I mentioned earlier, so this one cannot take on their own. So they have to come with a this TRF, that is this form from the medical certified medical practitioner. So here I have listed some of the next generation sequencing based test under this oncology, neurology, and reproductive category. So under oncology category, we have testing for BRCA1, BRCA2 for breast cancer. Then we have comprehensive tumor panels. So that test whether there are some variants for comprehensive. So whether there are some variants for the development of tumor in your this genome, and also DPYD mutation analysis. So this is specifically for the one treatment, that is 5-fluorouracil based treatment. So that is to test whether that patient is okay to have this 5-fluorouracil based treatment. In some patients, there will be this DPYD mutation. So in such patients, this 5-fluorouracil treatment won't be not effective, rather that will be toxic. So in most of these oncology doctors, they prescribe this test before this 5-fluorouracil based treatment. Then again, this JAK2 analysis and lung cancer. There are lung cancer panels, this hereditary non-polyposis colorectal cancer gene panel, and there are various other tests also. And under neurology also, there are this Charcot-Marie tooth panel testing is there, and to check the muscular dystrophy, whether there are this muscular dystrophy associated variations in the genome. So we have some panels for that one. Then epileptic encephalopathy gene panel. Likewise, there are a lot of panels. And for reproductive, mainly people test for this NIPT, that is non-invasive prenatal testing, to check whether there are aneuploidy or this genetic this issues present in the this gestational age, gestational offspring. Then also for this carrier screening. So these are actually, I have listed very few this tests. There are a lot of this 300 plus genomics tests available now, and people, this hospitals are this referring also, recommending also these genomic tests to have a proper this treatment guidance. And coming to this genomics testing technology, I think this I'm taking a lot of time, so now I will this move in a faster pace. Okay, so this genomics testing technology in our company, we adopt the genomic testing technology in this way. That is, we do the DNA sequencing followed by variant analysis. So DNA sequencing, as I mentioned earlier, this is next generation based testing or microarray based testing, and we do the variant analysis. And for this next generation DNA sequencing, there are these 4 major steps. That is, the first one is each of these part is also very crucial. That is, the first step is DNA extraction, and the DNA extraction should be proper. Then only we will be getting the proper output. And proper in the sense, the purity, the quality and quantity should be very good. Then after that, I'll go for this library preparation. This library preparation, that is a very laborious technique. Nowadays, there are automated techniques available for this library preparation to make it easier. And after this library preparation, then that is subjected to next generation sequencing. Then from the next generation sequencing, we get this data as a FASTQ file. FASTQ means, actually the details are given in the next slide. So first, the last step is the data processing or data analysis step. So we get the output of NGS sequencing as a FASTQ file. FASTQ file means actually we sequence some 20 to 30 base pair regions of DNA. So we get the details of that one as a FASTQ file, and when we do the data analysis, that is a very huge process. And for that, actually, this is a representation of that process. There are a lot of pipelines available for that. Most of the companies, they use their in-house developed pipelines for that. So once we get the FASTQ file, then we map that data in the file to the reference genome. So the reference genome, as we know, there as an outcome of the human genome project, we know what is our genome sequence is, and with that one, we do the reference genome mapping. And after the this read mapping, we get the this file in the VCF format. VCF means variant call format. So in that variant call format, so that there, if there is a base change, that is nucleotide change is somewhere, so that is mentioned, that is denoted in that VCF file, and there are specific IDs also for that, so that will be available in the VCF file. So once we get the VCF file, we go for the VCF annotation, that is the RSID. One of the IDs for representing variants is called RSIDs. So we go for RSID based annotations. Annotations means actually whether that variant is a clinically significant variant or not. And we need to, for especially for clinical genomics, we need to follow a certain guidelines, and this like ACMG guidelines, AMB guidelines, and all. So according to ACMG guidelines, the variants are classified into 5 categories. Those are pathogenic variants, likely pathogenic variants, variants of unknown significance, benign variants, likely benign variants. So like that. So if the variant is a pathogenic variant, that means actually so that is a risk case, and that is to be addressed properly. So benign and likely benign, they are they are harmless variants. They won't be doing any harm to that individual. Okay, so this is the place where we need a lot of this computational activities, and the skilled bioinformaticians, they have a role here. Okay, then coming to the last part, that is opportunities in genomics, and especially this is the overall opportunities. That is I discussed about the global market and this global key players and all. So this is the overall opportunities in this genomics field. So as we discussed earlier, so this has applications in various domains like healthcare and clinical diagnosis, pharmaceutical and drug development, and so on. Now, actually, what are the opportunities at OhMyGene? So you would be more interested to know about that one. So we have a lot of in-house research activities going on, and also a lot of this product development activities going on. So there are a lot of what are the opportunities there. So we can discuss that over here. That is variant analysis tools and pipeline development. As I told you in the previous slide, a lot of this computational power and bioinformatics tools and these technologies are required for this variant analysis. So we develop our own pipeline. So we have a pipeline, we are upgrading that pipeline, and for that actually we need the skilled bioinformaticians and computational biologists. And for also for variant prioritization pipelines we develop. That is when we get lots of variants, actually we also need to classify or prioritize that variant, and for that we also need to follow certain guidelines, as I mentioned earlier. Based on that, actually if we do all this manually, that will consume a lot of time, and that is not feasible for an industry. So for that we develop pipelines to automate all these things. So there this we require this people from our postgraduates from computational biology and bioinformatics. Then we do the genomics data research. Genomics data research means actually this has application in drug discovery. That is genetics based target identification. So this target identification is one of the key steps in the drug discovery. So which target is the suitable target to develop a drug? and whether that has a this genetics gene association for that target. So gene association can be in different ways. So we do a lot of research activity on that side. And also the gene expression analysis. Gene expression analysis means in some conditions there may be differential expression of certain sets of genes. That differential expression might be due to variations or some other associated genes activity. So what is that expression, differential expression present in a particular condition, and what are the cause for that gene expression? We do study that one. Here, this people from MSc biotechnology background, even MSc zoology, botany background, if you have a this interest to this one, you can also this pursue this. Then the next one is biocuration, and this for this biocuration, and this students from various domains can be this can opt this biocuration activities. That is, curation means actually very carefully we need to identify, we need to might be through this, we need to search through a whole lot of databases, lot of publications and all, and we need to curate desired information. So mainly to develop databases for the this industrial applications. So here if we say curation means actually we do curate variants for conditions. That is actually for some, suppose say condition A, breast cancer. What are the this genes and variants associated with that? So if we have that information, we can also develop our products. That is panel based, gene panel based product. So this is the objective of this biocuration and the database development activity going on at our center. Then also we have this nutraceutical development activities. Nutraceutical development means actually, as I mentioned earlier, this Ayurveda has a very this big role in this personalized medicine. So we are also exploring the potential of botanicals or Ayurveda related medicine and also nutraceuticals through this as the proper scientific research and validation. So that also people with this background, this botany background or this interest in this Ayurveda, they can also pursue this field. Then we develop this molecular biology based assays. For example, to confirm this gene variation, we develop real-time PCR based assays and also Sanger sequencing based assays. So people with a molecular biology background or skills, so they this area will be suitable for them. Then, as I mentioned in this biocuration, we do develop gene panel based products. So these are our this in-house research and opportunities going on at our center. And actually, yeah, so for all these disciplines, people, the students from all these disciplines, they can explore all these activities. And so there are a lot of job opportunities are also in this field. So even though in Kerala we have limited options, outside Kerala and this outside India, there are a lot of opportunities in this genomics field. So, so this actually, I just want to mention this news article. This came came last week in the Hindu newspaper. So this is about the one report. This submitted by Kerala State Higher Education Council. So they stated that, so here I have this marked this region. Here they stated that despite the increase in research output seen over the last few years, the assessment flagged the untapped potential of academia industry collaborations. So yeah, still that is a major problem actually I have seen in Kerala, but in outside Kerala and all. So people are exploring this academia and industry collaboration a whole lot. But in Kerala, we are still to tap that this industry academia collaboration. So there is a potential. Even our authorities are also this realizing this the need for this industry academia collaboration. And Dr. Gauri will be talking more about this one. And so with this, we invite this all of you to embark on a journey of learning and discovery at OhMyGene. So we can this do a lot of this good things together. And you are all young minds with a lot of ideas and everything. So you can come discuss your ideas. We can develop good projects together, and we can also work together. So with this, I conclude this session. Thank you all, and I'm happy to answer questions.
Thank you so much.